genomic data as well as the outcome data from the real world patients only enforced, my belief that genomics isn't just possible, but it's essential.
Hello, and welcome to the Veterinary Cancer Pioneers Podcast, the show where we delve into groundbreaking work of veterinary professionals dedicated to advancing the field of veterinary oncology. I'm your host, Dr. Rachel Venable, and I'm thrilled to embark on this journey with you. Today's episode is brought to you by Impromed, pioneers in AI-driven precision medicine for veterinary oncology. Their personalized prediction profile helps you make confident treatment decisions for your canine lymphoma and leukemia patients by predicting how your patient could respond to multiple chemotherapy protocol options. Learn more at impromedicine.com. That is IMPRImedicine.com. Welcome to the Veterinary Cancer Pioneers Podcast. I am so excited today to interview our guest, Dr. Guannan Wang. She is the founder of VetOmics. Dr. Wang brings over a decade of expertise in the canine genomics and precision medicine space, and she has a strong focus on advancing cancer care for companion animals. And just to give you all a little bit more background on her, she has been at the University of Pennsylvania and was also at Venable Animal Health. She played a key role in bringing genomic diagnostics into routine cancer care for dogs. She's led the development of innovation genomic tools and precision oncology databases. And I guess she's personally overseen thousands of reports on genomics. So Deb, I can't even imagine. I struggle reading some of these. So she really understands this. And her research has made significant contributions to understanding the genomic drivers, especially of hemangiosarcoma, and has shown clinical utility of using genomic testing in guiding treatment decisions, supporting diagnosis, and improving patient outcomes. So really excited to dive more into this. This is a really new and advancing area in veterinary oncology. So Dr. Wang, thank you so much for being with us today.
Thank you, Dr. Venable, for the invitation. And it's such a great honor to be here today. I can hardly call myself a pioneer, so I was mostly standing on giants' shoulders. So I've been learning along the way with the whole community lifting me up. So I love to share what we have learned during this whole journey with you and your listeners. It's a great honor to be here.
Thank you. And I think you're being very humble. You are certainly a pioneer. And, I always like to start and just get to know more about you guys. So tell me, what got you into cancer genomics, precision, and then kind of how did you end up in the veterinary space?
Yeah, it's quite like a non-conventional, right? So a Chinese-faced individual entering veterinary medicine. So not too common, at least during my time when I first came to the States back in 2007. And my PhD training was in molecular and cellular biology, actually at ASU Tempe campus, very close to you. But I was always drawn to research that could have a more direct clinical impact. So this is why during my postdoc training at UPenn, so I had the privilege of working with my mentor, Dr. David Roth, who actually built one of the nation's first human genomic diagnostic labs. It's called the Center for Personalized Diagnostic Labs back in 2012 to 2013. That experience really opened up the door for me to enter the field of cancer genomic diagnostics. At the same time, both me and Dr. David Roth are hardcore dog lovers. So Dr. Roth, we saw a clear gap between human and veterinary cancer care, right? And clear unmet need. So that really motivated us to try and bring precision medicine into a veterinary space. Also, very, very selfishly, so at the time, this was also a kind of career opportunity for me. So with our PenVet collaborators, Dr. Nicola Mason and Dr. Amy Duham, so we published some of the early studies on canine hemangiosarcoma genomics and its potential clinical relevance. So that's kind of really how I entered the space of veterinary medicine. I've been having fun ever since.
What ended up inspiring you to then go on past that journey and open your own genomics? So the VetOmics company, what inspired you to do that?
Yeah, so a lot of things, right? So the precision medicine was like taking shape back in 2012, 2013, like a lot of like a poster cancer types, right? Where see clear benefit indication where genomic transform the clinical outcomes, such as chronic myeloid leukemia, CML, breast cancer, melanoma, lung cancer, colorectal cancer, all these poster cancer disease. But for me personally, when I was invited to speak at AMP, American Molecular and Pathology Annual Conference, a huge conference, and AACR, American Association of Cancer Research. So this is almost the human equivalent of a VCS in our vet space. And back in 2019 and 2020, it was pretty striking to see that study after study to showing that genomic guided cancer care really improved the prognosis and survival. Granted, by 2012, a few years back, this conversation has shifted from, we saw all these benefits to more nuances, like resistance mechanism, right? Choosing the right drug, choosing the right patient, and the timing. So it's not simple like, when you apply genomic guided treatment, it always works. There's so much nuances to that. But to me, an overall trend at those conferences, the question was never, should we do this or should we not do this? It's a question that, how should we do this? What takes it to do it well? So that really like striking to me, right? So, okay, this works, but it's complicated. It has a lot of nuances. So after I left Penn, so I joined VDM and I had the opportunity to work with an outstanding team, including Dr. Will Hendricks, Dr. David Howard, the amazing team. So we actually brought genomic diagnosis into routine practice. So that was the early experience, right? But for me personally, whether I was at Penn or at VDM, so I was always being in the thick of the data. So seeing firsthand the genomic data as well as the outcome data from the real world patients only enforced my belief that genomics isn't just possible, but it's essential. This is combined my experience in the human space. So you ask about what makes us to finally decide to launch our own service, VetOmics. So there was also quite a story, but the bottom of it is that the gap of the knowledge was always from the center of my mind. We have the biggest gap in knowledge. The human precision medicine and the genomic guided or omics guided, it didn't happen overnight. It's after sequencing millions of cancer samples, right? And to accumulate that much of knowledge so that you can apply that knowledge back to practice. So of course, we are privileged, right, to have the human as our proof of concept, right? We can learn from that, but we cannot assume that it translates perfectly, right? So that really that gap of knowledge is, always kind of, I don't want to say this in a negative way, but bothered me that most of the cancers, we're lacking comprehensive genomic understandings for most of the cancers. And the ones that are studied are often studied through the lens of equivalent human cancers, which don't fully capture what's unique in dog tumor genome. And that knowledge directly translates into practice. So it's not just the biology, it's not just science that also affects the clinical practice. Now that we have multiple narrow human-inferred panel-based genomic tests, sometimes yields empty results, right? It's not ideal. And even if when there is a positive hit, there's always that nagging feeling, can this be the real driver of this cancer? Because the most effective way is to actually target the driver of the cancer so that it will respond the most effectively, right? And second of all, like this is a lack of genomic data. And second of all, lacking primary clinical data. I think this is even more, painful for you guys as a clinician than for me as a genomic scientist, because I'm not dealing with patient care like day in and day out. I'm dealing with mostly data. But, like a lot of these biomarker drug associations that come from human side, human influence, right? While again, we are lucky to have that rich human knowledge, we cannot just take it and assume that it works. So that's also another gap that really motivated us to start VetOmics. Granted, there are other bigger things that we need to figure out as a community, like access to drugs, right? Access to better, more drugs. Right now, we have a handful on a human space. They have hundreds of them. So, but our mission is to deliver the most comprehensive genomic insights through the highest quality testing, so that we can at least start off with the right genomic insights to guide both clinical practice or drug development when we down the line. So that's where we come from.
Well, that sounds amazing. Those are great goals. And I love how you said with genomics, it's not, the question shouldn't be should we do it or not. It should be how. How do we do it? And I think that's so true. How do we use this? How, what is the best? And so you were diving into this. What would you say with knowing, like with the genomic sequence, so like doing VetOmics, how would you say that those results will help with, diagnosis, therapy, selection, prognosis? What do you see as like the most helpful, or kind of where you're focusing? Because I know you mentioned like we need to still learning about some of these different areas, but where do you see it being the most effective?
Right now, therapy for sure. We know that currently genomics is, at the core of almost every aspect of cancer care in people. But we are still quite young in that development phase. So right now, the primary role of genomic testing is treatment selection, is to guide therapy choices. So with our comprehensive genomic profiling, so we can provide treatment recommendation across targeted therapies that are available in the vet space. Immunotherapies, we are lucky to have them, and chemotherapies and radiation therapies. So really, therapy selection or stratification is the primary role, and that's what the doctors use us for, right? So, yeah, and but that being said, we have been seeing an increasing utility of genomics in the diagnosis as well, especially in those challenge to diagnose cases where genomics actually can pinpoint which one is it in a like otherwise equivocal case based on cytology, based on histology. So we have 1/3 of those cases that could benefit from genomic testing. Yeah, even diagnosis. Yeah, prognosis right now. So most of the prognostic biomarkers are come from humans. So I make it very clear, just because we find a prognostic biomarker that predict poor outcome in the human cancer doesn't mean that we should stop treating the dog. Doesn't mean we need to really evaluate this in the canine patient, primary patients. Yeah.
It's interesting you mentioned diagnosis, because I remember hearing a pediatric oncologist once, and she was talking about how they sequenced every tumor. And that's really helped them, especially with some of the more rare types, but that they always do sequencing. And that just kind of opened my eyes, because I was like, oh, we don't do that.
Yeah. Not yet. We don't.
And so you mentioned, like trying to figure out like the driver, like what could be the driver of the mutation and the cancer essentially. Are you guys looking into trying to figure that out? How do we figure that out? How do you go about? Because I agree with you, that is the ultimate question, but how do we answer that?
So this is my job is to figure out. We have a lot of databases, right? We have a lot of AI assisted prediction algorithms. So after all that annotation and interpretation, it still comes down to what do we think is the primary driver for this particular tumor, right? For this particular dog. And there's many steps to kind of prioritize that. The first, this is getting into the weeds, but I love it. So like whether we have, for example, allele frequency of variant, whether it's 60% or it's 3%, that makes a huge difference, right? So even if we find a traditionally oncogenic mutation, well-established oncogenic mutation, but it's only present in 3% of the cancers, of the tumor cells. And while this tumor, the overall tumor purity is 90%. So do you think this is the main driver? Probably not, right? That's why, a lot of these, like a BRAF trials, it didn't work as it was expected, probably because the BRAF, although BRAF is present, but it's not the main driver, and that there are like a level of copy number changes, right? Whether this is a 38 copy number gain or this is a 2.5 copy number gain, we would definitely prioritize the higher copy number gain, right? And before that, so we have to find what are the potential cancer genes, right? This is where, we can really leverage the human databases, right? So that although I do want to say that we should leverage that, but also don't overlook the fact that we may be able to find a novel cancer genes specific for dogs that have not been defined as cancer genes in human cancers, right? So it's a complementing each other process. And hotspot mutations, so this is all be taken care of by the databases, by the algorithms, right? And at the end of the day, you have to figure out the level, what could be the most likely driver. And sometimes there are several, multiple, angles that we can target, which is, the best case scenario. I often say that, this case is full of potential clinical actionabilities. I think any of this therapeutic axis won't go wrong. You can, that also opened the door for us to combine different therapies. So we have quite some exciting data coming out. Yeah.
Really cool. And what would you say is often the most actionable finding? And does it often change how clinicians treat? I don't know how much you hear back from them, what they decide to do.
Yeah. So the big component, now that we feel like, okay, we got the genomic covered, so we are on the right foot of providing the best unbiased genomic insights. So the next thing we want to do is we don't have a lot of large-scale prospective studies to test, drug versus biomarkers, right? So we kind of have to do that through what we call hypothesis-driven clinical studies. So we hypothesize that the human-inferred drug biomarker associations, it provides a foundation for us to treat, right? But we really have to follow that. But in that framework, we have to conduct outcome research, follow up with the patient, and see whether the genomic-guided treatment actually improve outcomes, right? So that's the essence of what we do. I know we are a genomic diagnostic company, but I don't consider that getting a genomic report from us is the end of our service. We need to follow up with our doctors, our owners, and really trying to get every dog that comes into our door is the data that we can and should learn from.
Were you collecting data then is that what you're saying? Yeah. So you're getting data.
Yes. We are getting outcome data from the patients, from the dogs that did P9CTP test and treated. Sometimes they are treated, sometimes they are not. So we can get to that later, but that's the overarching goal, right? We want to follow up.
Is it something that your goal is to see, how is it working? Especially if you do have some dogs that don't do it. That's kind of a nice control group in some ways because there are mutations and they did do it. So is the hope to not only help your research, but then publish it out to the community?
I have quite some publications there, so but I do think these are real data that can help feedback into how we treat, how we even recommend advanced cancer care. So but we say it real world, right? And we're not alone to put more emphasis. Partly it's by not by choice. We would love to have prospective studies, right? But the almost 10 years that I've been doing this, we haven't been able to do that. We haven't been able to, even launch a couple of them. So, but I think we have to really value and emphasize the value of real world patients follow up. And there's even on the human side, there's a new type of clinical trials. It's called pragmatica clinical trials. And it also shifts the conventional prostate trial where you have to have such stringent control, right? And all these different criteria as well right now. So we kind of open it up to patients, right? And this not only validate the efficacy of the drug, but also the effectiveness, right? In a very noisy, background, the population. But if the signal is, we see the signal in this group, so then it's real signal. That's the hope.
So are you guys working with any other groups like clinical trials or are there any, because I know you work with a lot of prominent oncologists. So I didn't know if any of those guys were running trials or what all you are up to.
We have quite few studies that are underway. Some we wrote grants for, and some we are collecting data stage. So quite a few of them. Spanning targeted drugs as well. But when I say targeted drugs, so I meant small molecule inhibitors. But I know this means very differently in a clinician perspective. Targeted drugs as well as immunotherapies. We are involved in a few of them. So hopefully right now, so some of them are post retrospectively, right? Where we see whether, there is always for any given drug, at least from what I heard, there's always these excellent responder groups. And there's always these all, a non-responder group whatsoever. And so we want to see, okay, so can we see any correlative biomarkers to stratify the two groups? And then from there, then we can design, prospective studies.
Right. I'll have to look out for those different studies for sure.
Yeah. We're relatively young. We launched not too long ago. But I'm so glad. I'm just so grateful for the trust and I know a lot of the doctors and partners have placed in us, and that really is something that we need to work really, really hard for.
So what would you say you're seeing, talking about the different treatments and things? Do you see genomic sequencing and the targeted therapies or the small molecule inhibitors as more of the main solo treatment? Or are you seeing that this works a little bit better if, let's say, you combine chemo and then add in a targeted or basically a multi-drug treatment? What are you seeing the better outcomes, if you can say that, if you're seeing a trend?
Yeah. So that was a learning curve for me too. So I love to share what I have learned. At first, being a genomic scientist in the data day in and day out, I would think, okay, so when we have this perfect driver, when we have this drug that can target this driver, the doctors, they have to treat with this. So abandon all the other treatment plans to just use it. That was kind of naive. Later, I learned that, it's always not that simple. What the dog was already on makes a huge difference, whether they respond or not. It's easy if it doesn't respond to previous treatment, and then you can kind of switch out, right? But most of the time it's okay, responding, stable disease. So at this time, but not quite, halt the progression. Okay. So a lot of doctors, they actually combine the target therapies or immunotherapies to the, standard of care chemotherapies. And so that's, really, I think, boost the survival curve, at least based on, like a small cohorts that we have followed up so far. So it really, the combination therapy, I think, right now is definitely, I want to say, the way to go. So if it's not a clean cut, it's not replacing standard of care therapy with the genomic guided therapy. It's complementing, adding in, right? So that you can, really push that survival curve out by a lot. And when I say, that's another thing is that when I say target therapy, in target therapy, I think we should kind of step back and really, so it's not just the target therapy, it's immunotherapy as well. It's anything that's basically biomarker-guided, right, therapy. A lot of the immunotherapy from the human side are also guided by biomarker predictions, right? For example, the famous Keytruda PD-L1 antibody, it's primarily used by stratifications of PD-L1 IHC, as well as tumor mutation burden, and sometimes in specific cancers, microsatellite instability, MSI. I think, when we talk about target therapy, we shouldn't, focus only the small molecule inhibitors. Immunotherapy is very well in this group that can be stratified and predicted by, genomic biomarkers.
That's a good point. I hadn't really thought of that. Really accurate.
Yeah, it's I think eventually the biomarker biomarker-guided treatment, which include small molecule inhibitors, immunotherapies, some chemotherapies, and radiation therapies. Very, very broad category.
Yes, very broad. And so what advice would you give oncologists that are considering adding in genomic testing? Because like we talked about, this is a newer field, something we're learning about. So how do you recommend people think about this or go about it?
By now, I think a few years into, so everybody at least have heard of it, right? So, but there's a lot of different voices, right? That, for example, the hesitation can come from, there's not enough data, not enough PK/PD, not enough clinical data, right? Which is all true, but we also should not wait for the primary prospective study to happen, right? In the meantime, we can do a lot of things. We can treat the dogs with genomic guided treatment and follow up and see whether this works, and learn from there and feedback into the cycle, right? So I think we actually, both anecdotally, but also by small cohort, that we published and others published, have shown that genomic guided treatment actually improves outcome. As of today, now, so we cannot say that we don't have any data. We don't have as much data as that exist for people, but we have accumulated quite some data. I hear this all the time. So the doctors actually text me pictures or call me, when the dog responded beautifully to a treatment, right? That really makes, my day every time. So I think we work for that. We want to see that kind of response for all of our patients. Hopefully, that's the goal. The second of all, there's also, I think, unknowns about the science, the biology. Do we really think that the human driver is also the driver in dog cancer? So I think for that's another thing. I think, of course, the functional validation in cell lines are critical to prove the functional impact of a mutation on, oncogenesis, right? But I think equally important is the context of the cancer, of the tumor genome, the background. Like going back to the example of just because you have BRAF mutation at 3%, it might not do anything. to your treatment, right? So it's really, what other co-mutations occur, how to compare everything, and what comes up on top as like a most putative driver, and then we treat with that. So this is also kind of a learning process. I learned from some of our patients, if we prioritize this drug, but it doesn't respond. So I was say, okay, so then in this cancer type with this mutation, then the dog doesn't respond in this context. So that's also, we kind of iterate, right? So what's the best, recommendation? What's the best prioritization for the next one?
Yeah, that is really interesting. And just like you said, I'm sure you have a crazy algorithm trying to figure out all this data. It sounds quite complex, which is great. I think that's good.
Yeah, it's getting more complex. We get to learn so much of the insights for the tumor genesis, right? The biology, but also drug resistance mechanism. We can see clearly, how this tumor tree evolve under certain drug treatment and come out escape that treatment, and they come out a new metastase to a new site, essentially a new tumor. So it's pretty amazing.
What have you found working on, both the human genome and the dog genome? Is the dog genome a little bit simpler because they're so inbred, or is it actually more challenging? Just kind of curious, the comparison between the two.
To say that they are comparable or not is oversimplifying. I think each cancer type, each individual can have very, very different mutation profiles, even the same cancer types, right? So it sometimes can be a high tumor mutation burden. Sometimes can be high copy number alterations, extensive. You can see this genome-wide. So we use some of these genome-wide features. Actually, they are also sometimes diagnostic. I don't want to, compare human, for example, breast cancer to canine mammary tumor. So we definitely see some of the more prominent drivers, right? But they also have a different background context, right? That's why when we publish, we tend to really trying to grab the best story, right? What is the best storyline? What is the story that can get us funding, for example? Or we don't tend to report, all the noise, right? There's a very clear storyline. How do we package this data, right? To say it's actually, now I see the data, it's not always canine mammary tumor has the PIK3CA mutations similar to human breast cancer. Definitely not even 50% of mammary carcinoma has the PIK3CA. So there is a lot more to that than those well-picked. I don't want to say cherry-picked because they are real, but they are over-amplified signal oncogenic mutations. Osteosarcoma, for example. So it's it doesn't only involve mutations in TP53, STAT-D2, right, and a few other epigenetic modifiers. It's so much more than that. It can involve extensive copy number alterations in double-strand DNA repair pathways. That sometimes can actually help me diagnose osteosarcoma, because when you see when the top differentiation of a equivocal case is osteosarcoma versus a hemangiosarcoma, then these genome-wide features can really push it more towards one another. So I guess the short answer to your question is that we definitely see a lot of similarities, right? And the famous oncogenic mutations or famous oncogenic signaling pathways that are parallel between human cancer and canine cancer. But there's also, a lot of cases don't have these typical signals, but they're still called the same cancer.
So, what do you think the impact of precision medicine will be on the conversation between veterinarians and pet owners? Like, what, especially when they're discussing treatment options?
I hope by now a lot of the pet owners, from their own experience, right, already know that the concept of precision medicine, right? So I think we now say that, okay, instead of, giving you the one size fits all standard of care therapy, like splenectomy, followed by doxorubicin, for a hemangiosarcoma, now we can try something different. We'll treat your dog based on the genomic profile that are identified in his tumor, and then we can combine that, or we can treat that single agent. And so I think it's really a evidence-based and a science-based approach. I would think that most owners would appreciate that, because as you guys know, you probably have more experience. You do it like first line, right? Just as I do it with the data, first line experience. Take hemangiosarcoma, for example. The disease come back a lot of times, right? Came back metastasized. So I think here, so instead of replacing, so we're adding in, we're complementing. Okay, so we can try new drugs, these new therapeutic options that might fit your dog's cancer better than the standard care therapy.
I definitely found a lot of owners are somewhat familiar with genetic sequencing as far as, even commercials on TV talking about some of the targeted drugs or, breast cancer, lung cancer. So I agree. I certainly think they have heard of it, maybe not always understand exactly what's going on, but I think it does help with the conversation.
Yeah, I hope so. And again, as we do more of the testing and we publish, so, and we, everyone that use genomic in all of their patients, they don't see benefit, then we need to have a conversation, whether we're doing it right or, what's the problem. But, I hope most of the doctors that use us see the benefit, at least for, a certain type of cancer. But from like a aggregated data point of view, so we actually see a pretty well represented cancer types throughout both common cancer, rare cancer. We hope that everybody can see benefit, and then this, goes on and a positive feedback, right? And we have to publish.
To do your test, what kind of tissue samples, what do you guys need to run this sequencing?
We can actually run our test on a wide range of samples. FFPE, so that's the most common diagnostic specimen type. FNA, cytology sample, fresh, frozen tumor, fresh tumor. So that's our favorite kind. And urine, a fusion, bone marrow, and blood for leukemia, like in full blast. So that's the common types that we have used so far. True cut biopsy. So we can have optimized the workflow for a lot of these different cancer types, and we have been so far we have not failed to generate a report for any of our cases. We have done over 300 cases now. Among these, one case need a second sampling. This is at the early days where we were still, optimizing the workflow. And we have 19 cases that need a second workflow attempt, like a library prep, redo library prep, or resequencing. But overall, I think the yield is very, very high. Quality is very high as well.
Samples that needed the additional, were those cytology samples generally?
The one sample that need a second sampling is at first, naively, I was thinking, oh, okay, let's do FNA liquid instead of FNA slide. So FNA slide is a very standard sample type for genomic testing, right? And the viability is turned out much better than the cells in liquid. So the cells don't survive in liquid for very long. So we learned that from that patient. Early days, but ever since we switched to FNA slide, we didn't have issues. Yeah. Sometimes we know like the intrinsic quality of the FFPE can be harsh on DNA, right? So sometimes we encounter like a very degraded DNA from a FFP sample. Even with those, we have we sometimes have to sequence a couple more times in order to get to the target coverage. But yeah, we've been able to do that, and really have really, at the end of the day, it's how much insight, right, that you can provide, that you feel okay, this makes sense. We're not missing anything.
This has been fantastic. I love hearing about all the differences with genomics and how we can use it. And before we wrap up, how can people hear about you or get in touch with you? Do you want to mention like your website, or how would you like, anyone listening to learn more?
So VetOmics, animalhealth.com is our website. And so we've been fortunate enough to have a lot of support from the community that we started our own podcast, Precision Medicine. And so where we, kind of having these engaging conversations with oncologists who have a lot of experience, and just talk openly about the ins and outs, challenges. That's the thing that I really want to focus on. What are the challenges that we use genomics today? And of course, the promise is that's what I like to hear, right? And like the successful stories. And really, so the goal is to I call demystify genomic testing, precision medicine, right? So we have a lot to learn, but I think we are finally on the right trajectory. That we can really, whether this will truly help, and what do we need to do to make it work.
Sounds like lots of information, great ways to learn more and to reach out to you guys and get more information. And so kind of last question as I wrap up, who is someone else that you would recommend for this podcast?
I've been following your podcast. I know you always, so you kind of already interviewed everyone that I want. I would recommend. So, but I don't think you have interviewed Dr. Barb Kitchell. She's so down to the earth, and she asked the most sharpest question, and really make us all think. She doesn't have to do what she does now, but she's still pushing the boundaries and trying to bring more drugs for dogs to use, dogs and cats. I think she's someone that I would recommend.
Yeah, those sound great. And you're right, we have not interviewed Kitchell, so that certainly is a great recommendation. So, well, Dr. Wang, thank you so much. This has been so insightful, and I've learned a lot. I know our audience will too. So thank you again for being on our show.
Thank you for having me.
Well, that's it for this episode of the Veterinary Cancer Pioneers Podcast. If you enjoyed this episode and gained valuable insight, we would be so grateful if you could share our podcast with your friends and colleagues. And it would be even more wonderful if you want to give us a 5-star rating, positive review, or any kind of feedback on Apple Podcasts or wherever you listen. The Veterinary Cancer Pioneers Podcast is presented to you by Infomed.